I would like to introduce the next speaker, Dr. Kinga Gzieło. She obtained her PhD on the Jagiellonian University in Krakow, Poland. She's previous scientific research was mainly focused on neuron-glia interaction during normal and pathological states of the brain. Right now, she is a postdoc in the Department of Behavioural Neuroscience and Drug Development at Maj Institute of Pharmacology. This is the Professor Valproic laboratory. So, Kinga, the floor is yours.
Thank you. So hello, my name is Kinga Gzieło. I work at the Department of Behavioral Neuroscience and Drug Development at Maj Institute of Pharmacology of Polish Academy of Sciences in Krakow. Today I want to talk about our recent data regarding autism spectrum disorder in rats. My speech will be divided into 3 parts. In first part, I will introduce you to ASD in humans and in preclinical models. Then I shortly remind you some information about USVs emitted by rats, and then I will talk about our data. Autism spectrum disorder is a set of neurodevelopmental disorders characterized by reduced sociability, communication, repetitive patterns of behavior, and some comorbid features like cognitive impairments. Although the number of ASD cases is still growing, there is no specific answer of what is the main mechanism of ASD development and how to treat symptoms of ASD. These answers are actually very difficult because of the complexity of symptoms of ASD and the variety of factors leading to ASD. There is no single unifying cause of ASD. Actually, the majority of cases are idiopathic, and here some genetic factors and environmental factors interact and lead to ASD development. Genetic factors associated with ASD development play a role in some processes in the brain, like brain development, neurotransmission, synaptogenesis, neuronal migration, and neuronal excitability. In addition, we have some environmental factors which may lead to ASD development. It is premature birth, maternal infection, prenatal exposure to some drugs, for example, thalidomide or valproic acid, and also advanced age of parents.
Excuse me, Kinga, we've got some noise. I don't know if maybe it's starting from your computer. Right now, it's not. When you are sharing, we've got some noises. I don't know if it's your computer because it's quite. Sorry.
Okay, I don't know why.
Okay. It's still, but if you cannot do, oh, okay. Right now it's okay.
Yeah, but I did not do anything.
It might be the problem with the microphones. Okay, okay. Thank you.
Okay.
Sorry for this.
Symptoms of autism spectrum disorder appear in early childhood and persist to adulthood, often having a negative impact on everyday life. These symptoms are as following: persistent deficits in social communication, insistence of sameness, inflexible adherence to routines, stereotyped or repetitive behavior, movements, use of speech or use of objects, deficits in social emotional reciprocity and failure to reciprocal conversation, deficits in maintaining, developing and understand relationships, difficulties in making friends or lack of interest in peers, hyper or hyporeactivity to sensory input, or unusual interest of sensory aspects of environment. Deficits in non-verbal communicative behavior, for example, body language abnormalities, highly restricted fixated interest, but of abnormal focus and intensity, and deficits in social interaction with others. Autism spectrum disorder is, of course, also associated with some structural and anatomical changes of the brain. Neuropathological studies are limited, but they have revealed some differences in cerebral connectivity, focal disruption of cortical laminar architecture, gliosis, altered amount of neurons and synapses, disrupted permeability of the blood-brain barrier, and brain overgrowth in children. Of course, there are also some sex differences in autism spectrum disorder. Males are more susceptible to ASD development. The male-female ratio is 3:1, not 4:1 like it was previously thought. Moreover, males have more externalizing behavioral problems like reduced prosocial behavior, but attenuated aggressive behavior, hyperactivity, and repetitive behavior. In contrary, women have more internalizing behavioral problems like anxiety, depression, and other emotional problems. Moreover, women are more likely to camouflage their symptoms, especially this regarding social behavior, and that may lead to being misdiagnosed or diagnosed too late. Because of the growing number of ASD cases, there have been developed some preclinical models based on factors which may lead to ASD development, and we have drug-based models, inflammation-based models, and genetic models. I will briefly talk about poly-IC model, which is inflammation-based model, because several studies have shown that maternal immune system activation may lead to ASD development in children. So to study the mechanism behind the maternal immune activation and ASD occurrence in humans, pregnant rodents have to be injected with poly-IC, which may lead to viral-like inflammation in offsprings. From our studies, I can tell that this injected with poly-IC rats have several socio-communicative features of autism, like impaired social behavior, impaired communication, repetitive behavior, altered levels of cytokines and neurons in the brain. About 15% of ASD cases have a genetic component, and the most widely used animal model of ASD here is fragile mental retardation knockout mice. These mice exhibit disruption of the morphology of the brain, hippocampi, cortex, and cerebellum, and also disrupted structure and morphology of dendrites and synapses. All of these models lead to some behavioral impairments like impaired social behavior, hyperactivity, stereotypic behavior, repetitive patterns of behavior, and also deficits in communication. Of course, there are some brain changes in preclinical models of ASD, for example, disrupted synaptogenesis, altered number of interneurons, especially parvalbumin and calbindin-positive neurons, and imbalance between excitation and inhibition. In our study, we used the valproic acid model of ASD. Valproic acid is an antileptic drug and mood stabilizer. It may regulate gene expression and modulate neurotransmission. However, it has been shown that VPA consumption during pregnancy is associated with an elevated risk of ASD development in offspring, in children. given this link between VPA consumption during pregnancy and ASD occurrence in humans, VPA exposition has been proposed as an animal model of autism spectrum disorder. From several studies, we know that valproic acid exposition may lead to reduced play behavior, reduced sensitivity to thermal pain, to reduced exploratory activity, and also attenuated anxiety and impaired social communication. We know that social communication can be objectively measured in preclinical researches because rodents are able to respond and emit ultrasonic vocalizations. These ultrasonic vocalizations cannot be directly compared to human communication, but changes in ultrasonic vocalization emission may serve as a readout of communication deficits in laboratory conditions. I know that it was already taught, and I think it will be taught a lot of times today, but I want to quickly remind you about rat vocalization. Rat vocalizations can be divided into three main groups: 40 kHz calls, 22 kHz calls, and 50 kHz calls. The 40 kHz calls are associated with negative emotions and are emitted by pups when they are separated from their mothers or litter mates. These 40 kHz calls can be divided into three main groups: shorts, flats, and frequency modulated calls. Then we have 22 kHz calls, also known as alarms, emitted by juvenile and adult rats. And these 22 kHz calls are also associated with negative emotions caused by fear or anxiety. But these 22 kHz calls may also serve as defense mechanism informing about presence of the aggressor. Then we have 50 kHz calls known as happy calls. Of course, they are associated with positive emotions caused by food consumption, social interactions, reward, or tickling. This group may be divided into, again, three main groups: shorts, flats, and frequency modulated calls. And these frequency modulated calls can be further divided into some subtypes like trills, ramps, steps, complex, and others. In our experiment, we measured some USVs parameters. It was average duration of USVs, frequency, peak frequency of the calls, the complexity of the emitted calls, and of course, number of USVs. VPA model of autism spectrum disorder is one of the most widely used animal model of autism spectrum disorder, but there is little known about communication deficits in this model. So we perform a longitudinal study to characterize these social communicative deficits in this model. Here we recorded pups during the maternal separation test, young rats during social play test, and adult rats during social interaction test. All of the experiments were carried out on normal as well as VPA exposed rats. And as I said before, females are at an elevated risk of being misdiagnosed, and most of the studies, of the preclinical studies, have been done on male rats. So we chose to perform all of the experiments on both sexes of rats. We used, of course, valproic acid model of ASD. So pregnant rat dams were injected with VPA on gestational day 13. This is the time of neurogenesis when neural tube has closed and neuronal progenitors are migrating and proliferating. So this is a very sensitive time of neurogenesis, sensitive to any environmental insults, which may lead to neurodevelopmental disorders like autism or epilepsy, for example. Then on the offsprings, we perform maternal separation test on postnatal day 9. Each of pups was separated from its mother, placed in a special box and recorded for 5 minutes. Here you can see that we did not see any sex differences between pups, but we observed that BPA reduced the number of emitted calls, average call duration, and caused the increases in the peak frequency of USVs, but had no effect on the frequency modulation of calls emitted by pups. So we observed not only quantitative, but also qualitative alteration of USVs emitted by pups. We also looked closer to the distribution of the calls of the frequency modulated flats and shorts, and we did not observe any changes, but as you can see, the complexity of distributed calls are rather simple with prevalence of flat calls. Actually, this is normal because complexity of the calls develops with pups' age. So we might assume that all the rats will have more complex calls than pups, and you will see that in a few moments. Then on postnatal day 30, 31, 32, we perform social play test. Social play is also known as play fighting and refers to very vigorous and highly rewarding interaction between rats. During the social play, rats emit a lot of happy calls. And here we score 2 active social behaviors, pinning and pouncing, which are very specific to social play behavior in rats. Here we, in line with other studies, observe sexual dimorphism in juvenile play behavior because VPA reduced a number of pouncing and pinning, but only in VPA exposed male rats. It also led to reduced total playtime duration and also in male rats. This VPA also reduced the number of emitted calls and reduced average call duration, but in both sexes of rats. And VPA caused the increases in the peak frequency of USVs, but had no effect on the frequency modulation. So similarly, like it was in pups. We did not see any changes of social play behavior in VPA female rats, but female were actually less engaged in the social play. So we cannot exclude that relatively low number of social play episodes didn't allow us for demonstration of potential deficits in VPA female rats. Then on postnatal day 65, we perform social interaction test on adult rats. Here, 2 unfamiliar rats were of the same sex, and treatment were placed in the open field arena and recorded for 10 minutes. During the test, we scored following active social behaviors. It was sniffing, anogenital sniffing, following, mounting, and social grooming. Here, sex didn't affect social interaction. Actually, females and males were comparably engaged in social interactions, but we observed that VPA males have reduced number of social behaviors and reduced time spent on social contact. The diminished social behavior was also accompanied by reduced number of emitted USVs, also only in VPA males, but VPA had no effect on the pulse duration, on the peak frequency, and on the frequency modulation. So we observed that VPA, prenatal exposition to VPA, may lead to some social and communication impairments, which can persist into the adulthood. Moreover, we also looked at co-types characteristic, and here we observed that VPA juvenile rats emits more trills than normal rats. These changes of co-distribution may reflect an aberrant organization of social play behavior in juvenile rats, as specific USV types are associated with specific social play behavior. Then we look closer to the acoustic characteristics of emitted trills, and we observe that juvenile rats exposed to VPA emitted trills of higher peak frequency, of shorter duration, and their trills were not as complicated as these emitted by normal rats. Given that trills are emitted mostly before social contact, their higher number and aberrant characteristic may suggest that those trills weren't able to induce proper engagement in social play behavior in VPA exposed rats. We did not see any of these changes in adult rats. In conclusion, VPA exposure influenced social behavior of adolescents and adult rats, but the social behavior was changed mainly in VPA male rats. VPA exposition also leads to reduced number of episodes of social behavior and reduce the time spent of social contact. Moreover, communication deficits were observed during infancy, adolescence, as well as in adult animals, and the degree of VPA-induced alteration had the most pronounced impact on juvenile rats. What is worth to mention, altered acoustic call features are an essential element of an autistic-like phenotype, which you were able to see when I was talking about changes within trills emitted by juvenile rats. There are also sex differences in ultrasonic communication because females emitted less vocalization than males. And last but not least, ultrasonic vocalization measurement could be useful in monitoring lifelong social and communicative disability in Autism Spectrum Disorder model. I would like to thank my colleagues from my laboratory, especially Agnieszka Nikiforuk, who designed this study, and thank you for your attention.
Thank you very much for your presentation. Are there any questions from the audience? Okay, no one is actually raising their hands, but I actually do have one question.
Okay.
As this social behavior changed mainly in male rats, so that is, would you confirm that is actually corresponding to those differences in symptoms that both sexes show, right? Because you showed on the first slide, on one of the first slides, that there are quite some differences of symptoms that females show contrary to males. So does that reflect those different symptoms, or is it additional, but difference in rats, however, maybe not that corresponds to those differences in humans?
Maybe, but it is difficult to show these differences in rats. But as I said, female rats are less engaged in playing behavior. So maybe this may lead to that we cannot demonstrate differences between female rats because they play too little.
Okay.